It's not just the lipid bilayer. You've got embedded proteins of all sorts, but you've also got these channels and carriers and stuff that are going to make the membrane more permeable to certain things by allowing certain things through. Right? So this gets us into our whole transmembrane protein stuff today, right? And the basic idea is, although these lipid soluble molecules can get through easy, if you are a red molecule, you'll need this red channel to be able to pass through the membrane. If you're a green molecule and there's no green channels, you're not getting through the membrane, right? So all these channels are going to be allowing some things in, but they're not just going to allow anything in, right? So we'll look at the different types of channels today. And so we're going to break this down. You could look at these, you can categorize all these different channels in different ways. And we're just going to do it this way, that there's these passive and active processes by which molecules are going to move across the plasma membrane. And you could build yourself a little conceptual flow chart like this as far as transport across the membrane. You could break it into two big groups, active and passive. Right? And passive is going to include that simple diffusion, right through the lipid bilayer. And it's also going to include facilitated diffusion, right, of different types, all which are going to be protein mediated. And protein mediated also is involved in these active ones. And these are going to be either direct, so-called direct or primary active transport, or secondary active transport, as far as these protein mediated ones. There's also active processes here that are much different from this, which we'll go over, right? These vesicular transport. As far as breaking it down into passive and active processes, we want to always remember for these passive processes, we're always going to be going from a high concentration to a low concentration, right? Always going to be going from that high to low, right? Whereas this direct, in particular, you're actually going to be going against the concentration gradient, and that's why you need energy, right? Which is what this active thing means, right? And so you're going to do it, you're going to go from a low concentration to a high concentration, and you need to expend energy to do that. Once you do that, there's also a way, once you've built up this concentration gradients, you're going to use those concentration gradients to move stuff against its concentration gradient, right? So this is going to be this indirect or secondary ones. You're going to be moving things from a low or high concentration, to a low to a high concentration, but it's going to be coupled with this basically sodium ion flow from a high to a low concentration, just like diffusion. All right, we'll talk about this and go over everything there. All right, and then there's this other thing, which is, still requires ATP, but we don't really think of it in terms of concentration so much. For this passive part, again, you always want to think concentration. Everything's going to be moving down passively. You don't need to expend any energy. This is what they're doing naturally. You just got to let them, just give them a way to get in the cell, and they will flow in. Right? So all this, because it's not crossing right through the plasma membrane, it's called facilitated diffusion, right? And these are these molecules which are too big, they're too polar, they're ions and stuff, they can't move through that. And so they got to go through a protein channel. And we talked a little bit about the structure of them, but basically there's these big blobs here that are going to let in some stuff, right? Based on their concentration gradient. And there's two kind of general ways, which we'll discuss. Over here, they're either going to be a channel or a transporter, right? And there's a couple little differences here, which we'll touch on between the difference between these two. And so one of those channel mediated facilitated diffusion are those aquaporins, that is aqua meaning water, porins meaning pore. So these water channels, water can diffuse through the lipid bilayer, but mostly it is going through these aquaporins. So osmosis, which is that movement of water toward a higher salt area, is going to be going through these aquaporin channels. And again, it's a little polar molecule, so it'll go through these channels much easier than it will through the lipid bilayer, right? And so for any cells who are producing these, body fluids right there, you need a lot of water. So they're going to import a lot of water. Right? You're also going to control the concentration of urine in this manner. So aquaporins are going to be everywhere in the whole body. There's all sorts of different aquaporins. These are just basic channel mediated facilitated diffusion, right? They're going down there, water, it's doing osmosis, its own concentration gradient toward the higher solute level and toward a lower water level. It's going to be moving through these channels right here. And then the other main big kind of category of the ones we'll run into quite frequently are these ion channels, right? Sodium, chlorine, potassium channels. Big ones are the sodium and chloride channels. And this is like basically they're going to let these ions move down electrochemical gradients, because they can't pass through the lipid bilayer, right? And the amount of any particular channel in any particular cell is going to the permeability of that cells to this ion. So most cells have a lot of potassium and chlorine channels, right? And they're going to move down their electro concentration gradient. So one thing out is for some reason, and we're going to guess it's just due to a lot of these these kind of negatively charged proteins, maybe the phosphins. Cells have a kind of negative, overall negative charge without considering anything else. There's a negative charge, right? That is, there's more negative ions or negative parts of proteins or whatever, so that positively charged molecules would flow in and negatively charged molecules would flow out down their electric gradient, right? Which means if there's a lot of potassium channels in here, that means potassium is generally going to be flowing in toward this negative source because they're a positive ion, right? And that's what they'll do. I know this shows it going out, but this looks kind of opposite, but the leak channels, potassium is going, this I guess is showing out its concentrating in gradient, but the reason there's a lot of potassium in here is basically they're flowing in and out, they're flowing in toward their electric gradient, out toward their concentration gradient, right? They're leaking, they're going in and out based on whatever's happening in the cell, right? Sodium, whenever it could find a channel, will go into these leak channels. The cell is a lot more permeable to potassium just because you got all these much more leak channels in there. There's also chlorine channels, and because the cell is negative, chlorine is negative, it'll be flowing out of the cell. So calcium and sodium, those positive molecules are generally there's not so many leak channels, so they're kind of kept low. That's one way they're kept low. And potassium is much more permeable. So those are just like leak channels, which are just letting things in, they're freely flowing in. A lot of the channels though, these ion channels and other channels are going to be what's called gated. That is, there's going to be some kind of control whether these are open or not, right? So this is potassium right here. And we said that the membrane was highly permeable to potassium. At any point, you can close, for some of these channel proteins, you can close that gate and all of a sudden you're going to increase your permeability to potassium, right? So even though these are going down their concentration gradient, you could just close the gate and all of a sudden alter the permeability, right? So all of a sudden, if you close it, the cell is less permeable, more potassium is staying inside the cell. If you open it, right, then they'll start flowing down their concentration gradient, right? Either way, whatever their concentration gradient is, if it's closed on one side, or if it's closed, then they can't leave. If it's right, so these gates can be regulated in a couple of different ways. They can be regulated, right, based on different things going on around the cell, right? So let's just look at a couple of examples that we'll run into. We're going to run into during the course, right, when we get to the nervous system. Right, we're going to see this action potential come down a nerve. Right, there's going to be this electrical signal there's a voltage change that happens. When the voltage change hits these voltage gated calcium channels, right, which are just channels that will open up for calcium, calcium is going to flood in because it's been kept at a high concentration out here, right? And so the signal was this voltage change that came down here, and all of a sudden you got this calcium rushing in through these channels right here. Right? So it's regulated by voltage, right? So that's an electrical impulse. Another one is maybe a ligand, right? In this case, the cell, once those calcium is released, this causes a chain reaction and the neurotransmitters are released. Over here, they connect to these green channels over here, which open these channels up, right? And these are sodium channels. Once these channels are opened up, then sodium, which has been sitting out here at high concentrations, is going to rush in the cell, right? So these are ligand-gated channels. There's all the other types of channels, such as mechanical sort of gated channels where you have pressure or sound waves and stuff are going to open them and open up these channels, right? So this is a way of signaling, having a signal cause this change in membrane potential, which we'll get into later, right? But these are all types of gated channels. Okay, those are just some examples, but you get the idea, right? They're regulated by one thing or another. Ligands, voltage, other things. Maybe a photon reaching the back of your photoreceptors in your eye will open up certain sodium channels or close them, right? So there's all sorts of things that will open and close these channels. These are just two examples. Right, so these are still just these ion channels. Then the other thing about, so that's your ion channel. Sorry, that was it for your ion channels, your channels. Next type of facilitated diffusion here are these carrier-mediated facilitated diffusion. And in this case, you know, it's a little less like a channel that just things just pass through it, and maybe a little more like kind of like an enzyme that like binds something and it changes its shape. And in this case, the changing of a shape is going to allow passage of a molecule down its concentration gradient, right, from one side of the cell to the other part of the cell, right? So this is that carrier-mediated facilitated diffusion as opposed to our channels, right? So again, we're moving thing down its concentration gradient. The solute is going to bind a certain region, binds to a specific carrier on one side of the membrane, and it's released on the other side of the membrane. So it's going from, in this case, the outside to the inside of the cell right there, right? And this is all based on concentration gradients. If glucose, in this case, is high out here, it'll be binding on this side and be released into the cytosol over here. If the concentration hypothetically was high of glucose over here, the opposite would happen, right? It would always go down its concentration gradient. All right, so these are transporters. And the higher the concentration out here, the faster that is going to go. Also, like enzymes, it only works to a certain point because this can only bind so many molecules at once. It could only go so fast. So at a certain point, you could have a lot of high concentration, but you don't get any faster activity, right? And this is called saturation or the transport maximum, right? At some point over here, there is a lot of concentration. It's happening very fast. It's happening very fast, right? The higher the concentration gets over here, but at some point it starts to flatten out like this, right? Because basically you've used up all your carriers, that is those spaces that bind the solute, right? And it just only works so fast, right? Simple diffusion and maybe channel ones are going to kind of go at this kind of linear rate, whereas the channel protein, the carrier proteins are going to kind of have this curve right here. They're going to go up very fast, but then they're going to kind of flatten out. And if you increase the concentration, it's not going to affect the rate anymore. So that's saturation of these carrier-mediated facilitated diffusion. And the other thing is what I wanted to mention about this was just like your gated channels can be regulated in many ways, right? These also can be regulated possibly in those ways, but here's one about the transporters here, right? About how you can actually regulate the selective permeability of a cell, right? In this case, to achieve glucose homeostasis, right? So in this example, you have these GLUT4 transport molecules inside the cell. And over here, not in the membrane, right over here. You got this glucose outside, glucose isn't coming in. And then if you have high level glucose, that's going to cause somewhere down the line, and the pancreas is going to cause the pancreas to release insulin. Insulin floats around the blood until it gets to this insulin receptor. This causes all sorts of magical signals to make this sort of secretory vesicle bind with the cell membrane and just add these GLUT4, these GLUT transporters into the cell membrane. And all of a sudden your cell is more permeable and these cells can uptake glucose, right? So these channels are there. They're going to go on concentration gradient. If they're not there, the cell's not permeable. If they are, then the cell is permeable. You want to increase the permeability, you just increase the number of channels. And in this case, you're doing it through insulin, right? Which we'll get into a little bit more on next time, right? But this is just the selective permeability and thinking about kind of how that might change, according to different situations. So this is passive diffusion. All that is passive diffusion, right? Things, water going through those aquaporins, the channel ones, ions and stuff like that going through those channels, and then those carrier proteins going from one side to the other as well, right? All this from high to low concentration. You don't need to expend any energy. All you need is the channels for it to happen.